Right, we started with the next topic of biological oxidation. Oxidation is defined as a loss of electrons and reduction as the gain of electrons. This may be illustrated by the interconversion of ferrous iron Fe2+ to ferric iron Fe3+. Fe2+ oxidation or reduction, where you have accepted or reduced, and to give Fe3+ with electrons would be reduction, without electrons would be the electron lost, and the oxidation is accepted by an acceptor, which is said to be reduced. Thus, oxidation reduction is a tightly coupled process. The general principle of oxidation reduction is applicable to biological systems. The oxidation to NADH to NAD+ coupled with the reduction of FMN to FMNH2 is illustrated. In the above illustration, there are two redox pairs NADH, NAD+, and FMN, FMNH2. The redox pairs differ in the tendency to lose or gain electrons. Redox potential, the E0. The oxidation reduction potential, or simply redox potential, is a quantitative measure of the tendency of a redox pair to lose or gain electrons. The redox pairs are assigned specific standard redox potential E0 volts at pH 7 and 25 degrees centigrade. The redox potential of some biologically important redox systems are given in Table 3. The more negative redox potential represents a greater tendency of reductant to lose electrons. On the other hand, a more positive redox potential indicates a greater tendency of oxidants to accept electrons. The electrons flow from a redox pair with more negative E0 to another redox pair with more positive EO. The redox potential EO is directly related to the change in the free energy. Standard redox potential E0 of some oxidation reduction systems: succinate, 2H, NAD, NADP, FMN, lipoate, FAD, pyruvate, fumarate, cytochrome B, coenzyme Q, cytochrome C1, cytochrome C, cytochrome A. Electron transport chain: the energy-rich carbohydrates, particularly glucose, fatty acids, and amino acids, undergo a series of metabolic reactions and finally get oxidized to CO2 and H2O. The reducing equivalents from various metabolic intermediates are transferred to coenzymes NAD+ and FAD to produce respectively NADH and FADH2. The latter two reduced coenzymes pass through the electron transport chain or respiratory chain and finally reduce oxygen to water. The passage of electrons through the ETC is associated with the loss of energy. A part of this free energy is utilized to generate ATP from ADP and PI. Mitochondria, the powerhouses of cell. The mitochondria are the centers for metabolic oxidative reactions to generate reduced coenzymes NADH and FADH2, which in turn are utilized in ETC to liberate energy in the form of ATP. For this reason, mitochondrion is appropriately regarded as the powerhouse of the cell. Mitochondrial organization. The mitochondrion consists of 5 distinct parts. These are the outer membrane, the inner membrane, the intermembrane space, the cristae, and the matrix. The electron transport chain and ATP synthesizing system are located on the inner mitochondrial membrane, which is a specialized structure rich in proteins. It is impermeable to ions, H+, K+, sodium+, and small molecules ADP, ATP. This membrane is highly folded to form cristae. The surface area of inner mitochondrial membrane is greatly increased due to cristae. The inner surface of the inner mitochondrial membrane possesses specialized particles that look like lollipops, the phosphorylating subunits, which are the centers for ATP production. Mitochondrial matrix. The interior ground substance forms a matrix of mitochondria. It is rich in the enzymes responsible for the citric acid cycle, beta oxidation of fatty acids, and oxidation of amino acids. Structural organization of the membrane is highly folded to form cristae. The surface area of inner mitochondrial membrane is greatly increased due to cristae. The inner surface of the inner mitochondrial membrane possesses specialized particles that look like lollipops, the phosphorylating subunits, which are the centers for ATP production. Mitochondrial matrix, the interior ground substance, forms the matrix of mitochondria. It is rich in the enzymes responsible for the citric acid cycle, beta-oxidation of fatty acids, and oxidation of amino acids. Structural organization of respiratory chain. The inner mitochondrial membranes can be disrupted into 5 distinct respiratory or enzyme complexes, denoted as complex 1, 2, 3, 4, and 5. The complexes 1 to 4 are carriers of electrons, while complex 5 is responsible for ATP synthesis. Besides these synthesis enzyme complexes, there are certain mobile electron carriers in the respiratory chain. These include NADH, Coenzyme Q, Cytochromes C, and oxidase. That was a diagram for that. The enzyme complexes 1 to 4 and the mobile carriers are collectively involved in the transport of electrons, which ultimately combine with the oxygen to produce water. The largest proportion of the oxygen supplied to the body is utilized by the mitochondrion for the operation of electron transport chain. Nicotinamide nucleotides of the 2 coenzymes NAD+ and NADP+, derived from the vitamin niacin. NAD+ is more actively involved in the ETC. NAD+ is reduced to NADH plus H plus by dehydrogenases with the removal of 2 hydrogen atoms from the substrate AH2. The substrates include glyceraldehyde 3-phosphate, pyruvate isocitrate, alpha-ketoglutarate, and malate. AH2 plus NAD plus gives A plus NADH plus H plus. Components and reactions of the electron transport chain. There are 5 distinct carriers that participate in the electron transport chain ETC. These carriers are sequentially arranged and are responsible for the transfer of electrons from a given substrate to ultimately combine with proton and oxygen to form water. NADPH plus H plus produced by NADP dependent dehydrogenase is not usually a substrate for ETC. NADPH is most effectively utilized for anabolic reactions, example fatty acid synthesis, cholesterol synthesis. Flavor proteins, the enzyme NADH dehydrogenase, NADH coenzyme Q reductase, is a flavor protein with the FMN as the prosthetic group. The coenzyme FMN accepts 2 electrons and a proton to form FMNH2. NADH dehydrogenase is a complex enzyme closely associated with the non-heme iron proteins, NHI or iron-sulfide proteins FES. NADH plus H plus FMN will yield a 7-NAD plus FMNH2. Iron-sulfide proteins. The iron-sulfide proteins, FES proteins, exist in the oxidized Fe3+ or reduced Fe2+ state. Listen, students, there are certain portions like glycolysis and related aspects that I'll be repeating with many topics because there's a lot of correlation with these. So that's why we give you a continuation at the start or end of the lecture. So please take notes on this and correlate as you study, right? Okay, continuing with the subject. Now, about half a dozen ferrous sulfide proteins or iron sulfide proteins connected with the respiratory chain have been identified. However, the mechanism of action of iron sulfide proteins and the ETC is not clearly understood. One FES participates in the transfer of electrons from FMN to coenzyme Q. Other FES proteins associated with cytochrome B and cytochrome C participate in the transport of electrons. Coenzyme Q is also known as ubiquinone since it is ubiquitous in the living system. It is a quinone derivative with a variable isoprenoid side chain. The mammalian tissue possesses a quinone with 10 isoprenoid units, which is known as coenzyme Q10, COQ10. Coenzyme Q is a lipophilic electron carrier. It can accept electrons from FMNH2 produced in the ETC by NADH dehydrogenase or FADH to produce outside ETC, example succinate dehydrogenase, acyl-CoA dehydrogenase. Coenzyme Q is not found in mycobacteria. Vitamin K performs similar functions as coenzyme Q in these organisms. Coenzyme Q has no known vitamin precursor in animals. It is directly synthesized in the body. Cytochromes. The cytochromes are conjugated proteins containing heme group. The latter consists of a porphyrin ring with iron atoms. The heme group of cytochromes differ from that found in the structure of hemoglobin and myoglobin. The ion of heme in cytochromes is alternately oxidized Fe3+, and reduced Fe2+, which is essential for the transport of electrons and the etc. This is in contrast to the heme ion of hemoglobin and myoglobin, which remains in the ferrous Fe2+ state. These cytochromes were initially discovered from the mammalian mitochondria. They were designated as cytochrome, band A, cytochrome A, band C, depending on the type of heme present and the respective absorption spectrum. Additional cytochromes such as C1, B1, B2, A3, etc. were discovered later. The electrons are transported from coenzyme Q to cytochromes in the order B, C1, C1, let me just magnify that, B, C1, C, A, and A3, the property of reversible oxidation, reduction of heme ion Fe2+, squabble of its high energy, movement to high energy Fe3+, present in cytochromes allows them to function as effective carriers of electrons and He. Cytochrome C, molecular weight of 13,000, is a small protein containing 104 amino acids and a heme group. It is a central member of ETC with an intermediate redox potential. It is rather loosely bound to inner mitochondrial membrane and can be easily extracted. Cytochrome A and A3. The term cytochrome oxidase is frequently used to collectively represent cytochrome A and A3, which is the terminal component of He. Cytochrome oxidase is the only electron carrier, the heme ion of which can directly react with molecular oxygen. Besides heme with iron, this oxidase also contains copper that undergoes oxidation reduction, Cu2+, Cu+, during the transport of electrons in the final stage of Hc. The transported electrons, the free protons, and the molecular oxygen combine to produce water. The next portion would be oxidative phosphorylation. The transport of electrons through ETC is linked with the release of free energy, the process of synthesizing ATP from ADP and PI, that's organic phosphate, coupled with the electron transport chain, is known as oxidative phosphorylation. I repeat, the process of synthesizing ATP from ADP and organic phosphate coupled with the electron transport chain is known as oxidative phosphorylation. The complex 5, as in figure 6 coming up, of the inner mitochondrial membrane is the site of oxidative phosphorylation. P:/O ratio. The P:/O ratio is refers to the number of inorganic phosphate molecules utilized for ATP generation for every atom of oxygen consumed. More appropriately, P:/O phosphate is to oxygen ratio represent the number of molecules of ATP synthesized per pair of electrons carried through ETC. The mitochondrial oxidation of NADH with a P:/O ratio of 3 can be represented by the following equation. NADH plus H plus half O2 plus 3 ADP plus 3 PI gives NAD plus 3 ADP plus 4 H2O. P:/O ratio of 2 is assigned to the oxidation of FADH2. Note that although yet to be proved beyond doubt, some workers suggest P:/O ratio of 2.5 for NADH and H plus and 1.5 for FADH2 based on the proton translocation. Sites of oxidative phosphorylation. Okay. These are 3 reactions in the Hc that are exergonic to result in the synthesis of 3 ADP molecules. The 3 sites of ATP formation of ETC are oxidation of FMNH2 by coenzyme Q, oxidation of cytochrome B by cytochrome C1, cytochrome oxidase reaction, each of the above reactions representing a coupling site for ADP production. There are only 2 coupling sites for the oxidation of FADH. It's P:/O ratio 2 since the first site is bypassed. Energetics of oxidative phosphorylation. The transport of electrons from redox pair NAD plus bar NADHE is a redox potential at minus 0.32 to finally the redox pair half O2 by H2O redox potential at plus 0.82 may be simplified and represented in the following equation. Half O2 plus NADH plus H plus gives water plus NAD2 plus. The redox potential difference between these 2 redox pairs is 1.14 V. which is equivalent to an energy 52 calories per mole. These ATP are synthesized in the EDC when NADH is oxidized, which equals to 21.9 calories. Each ATP is equal to 7.3 calories. The efficiency of energy conservation is calculated as 21.9 into 100 by 52, which is 42%. Therefore, when NADH is oxidized, about 42% of energy is trapped in the form of 3 ATP and the remaining is lost as heat. Mechanism of oxidative phosphorylation. Several hypotheses have been put forth to explain the process of oxidative phosphorylation. The most important among them, namely chemical coupling, conformation coupling, and chemiosmotic, are discussed below. Chemical coupling hypothesis, I think we just discussed this yesterday, but as a continuation, this keeps going on, so listen carefully and make the necessary notes. This hypothesis was put forth by Edward Slater in 1953. According to chemical coupling hypothesis, during the course of electron transfer in the respiratory chain, a series of phosphorylated high-energy intermediates are first produced, which are utilized for the synthesis of ATP. These reactions are believed to be analogous to the substrate-level phosphorylation that occurs in glycolysis or citric acid. However, this hypothesis lacks experimental evidence, since all attempts so far to isolate any one of the high-energy intermediates have not been successful. Conformational coupling hypothesis. Paul Boyer proposed this hypothesis in 1964 as a consequence of electron transport. A conformational change in the membrane portion is induced. This is believed to be responsible for the synthesis of ATP. The protein attains a high energy conformation due to electron transfer, which, when reverted back to random state low energy, results in the production of ATP from ADP plus pi. There is some evidence demonstrating conformational changes in mitochondrial membrane proteins. However, there is no conclusive proof that such changes may be actually responsible for the ATP synthesis. Chemiosmotic hypothesis. The mechanism usually proposed by Peter Mitchell in 1961 is now widely accepted. It explains how the transport of electrons through the respiratory chain is effectively utilized to produce ATP from ADP plus Pi. The concept of chemiosmotic process hypothesis is comparable with the energy stored in a battery separated by positive and negative charges. Protein gradient, the inner mitochondrial membrane as such is impermeable to proteins, H plus and hydroxyl ions. See, this whole thing works on this principle of osmosis. That's why it's called chemiosmotic hypothesis. And hydroxyl ions, the transport of electrons through EDC is coupled with the translocation of protons, H plus, across the inner mitochondrial membrane coupling membrane from the matrix to the intermembrane. The pumping of protons results in an electrochemical or proton gradient. This is due to the accumulation of more H plus ions, low pH, on the outer side of the inner mitochondrial membrane than the inner side. The proton gradient developed due to the electron flow in the respiratory chain is sufficient to result in the synthesis of ATP and ADP and Pi. Oxidation by reduction of OR loops or redox loops. It is postulated that ETC is folded into 3 oxidation reduction loops corresponding to complex 1, 3, and 4. Each loop is a coupling site and consists of a hydrogen carrier and an electron carrier. 6 protons are translocated for each electron pair transferred from NADH plus H plus to oxygen. The 3 oxidation reduction loops, along with the carriers of protons and electrons, are depicted in the figure below. The first pair of protons, 2H plus, are translocated via FMN, and the second 2H via Coenzyme Q. This is the diagram. I've shown it to you yesterday. And this is a continuation. There is some controversy regarding the carrier of the third proton pair designated as X in the figure 11. You see the X here. This is there's a little doubt there, controversy regarding the notations there, right? That is the intermembrane space, the inner mitochondrial membrane, then comes the matrix. So it goes through that, right? So take a screenshot of this. It's very important. And now going forward, some workers believe that coenzyme Q is involved here. Coenzyme Q is a lipid-soluble small molecule and can freely move across the inner mitochondrial membrane. A cyclic process referred to as coenzyme Q is said to be operated for translocation of protons at 2 OR loops. Enzyme system for ATP synthesis. ATP synthase present in the complex 5 utilizes proton gradient for the synthesis of ATP. This enzyme is also known as ATPase since it can hydrolyze ATP to ADP and Pi. ATP synthase is a complex system of complex enzyme and consists of two functional subunits, namely F1 and FO. Its structure is comparable with lollipops. The protons that accumulate on the intermembrane space re-enter the mitochondrial matrix, leading to the synthesis of ATP. This process involving ATP synthase occurs in three stages. 1. Subunit FO of ATP synthase translocates protons re-entry into the matrix. F1 subunit catalyzes the formation of ATP from ADP and pi. This is believed to involve in conformational change in F1. And F0 and F1 subunits coordinate with each other, and the disappearance of proton gradient with ATP synthesis. And the evidence in support of KORAH-BIOTECH hypothesis. Many experimental observations support the KORAH-BIOTECH hypothesis. The intact mitochondria membrane is responsible for oxidative phosphorylation. The inner mitochondrial membrane is impermeable to protons, H+, and other ions, OH, K, chlorine, etc. ADP can be generated by increasing the H+ ion concentration by adding acid on the outside of mitochondria. Certain compounds which increase the permeability of inner mitochondrial membrane into protons inhibit ATP synthesis, example 2,4-dinitrophenol. Inherited disorders of oxidative phosphorylation. It is estimated that about 100 polypeptides are required for oxidative phosphorylation. Of these, 13 are coded by mitochondrial DNA, mtDNA, and synthesized in the mitochondria, while the rest are produced in the cytosol coded by nuclear DNA and transported. Mitochondrial DNA is maternally inherited, since mitochondria from the sperm do not enter the fertilized ovum. Mitochondrial DNA is about 10 times more susceptible to mutations than nuclear DNA. Mitochondrial DNA mutations are more commonly seen in tissues with higher rate of oxidative phosphorylation. For example, central nervous system, skeletal and heart muscle, and liver. Leber's hereditary optic neuropathy is an example for mutations in mitochondrial DNA. This disorder is characterized by loss of bilateral vision due to neuroretinal degeneration. Remember that Leber's hereditary optic neuropathy, right, is an example of mutations in mitochondrial DNA. Inhibitors of electron transport chain. Many specific inhibitors of electron transport chain have contributed to the present knowledge of mitochondrial respiration. Selected examples of these inhibitions are given in figure 7. The inhibitors bind to one of the components of ETC and block the transport of electrons. This causes the accumulation of reduced components before the inhibitor blockade step and oxidized components after that step. The synthesis of ATP, right, is its phosphorylation is dependent on electron transport. Hence, all the site-specific inhibitors of ETC also inhibit ATP formation. The three possible sites of action for the inhibitors of ATC are identified: NADH and coenzyme Q, fish poison, rotenone, barbiturate, drug emitter, and antibiotic peotein A inhibit at this site. Between cytochrome B and C, antimycin A and antibiotic British antileucide BAL, an antidote used for and against war gas are the two important inhibitors of the site between cytochrome B and C. Inhibitors of cytochrome oxidase: carbon monoxide, cyanide, hydrogen sulfide, and acid effectively inhibit cytochrome oxidase. Carbon monoxide reacts with reduced form of the cytochrome, while cyanide and acid react with the oxidized form. Inhibitors of oxidative phosphorylation uncouplers. The mitochondrial transport of electrons is tightly coupled with oxidative phosphorylation ATP synthesis. In other words, oxidation and phosphorylation proceed simultaneously. There are certain compounds that can uncouple or delink the electron transport from oxidative phosphorylation. Such compounds, known as uncouplers, increase the permeability of inner mitochondrial membrane to protons H+. Thus, an uncoupler allows the electron transport but blocks the establishment of proton gradient across the inner mitochondrial membrane. The result is that ATP synthesis does not occur. The energy linked with the transport of electrons is dissipated as heat. The uncouplers allow, often at accelerated rates, oxidation of substrates via NADH or FADH2 without ATP formation. The uncoupler 2,4-dinitrophenol DNP has been extensively studied. It is a small lipophilic molecule. DNP is a protein carrier and can easily diffuse through the inner mitochondrial membrane. The other uncouplers include dinitrocresol, pentachlorophenol, trifluorocarbonyl cyanide, phenylhydrazone. And the last compound, FCCP, is said to be 100 times more effective as an uncoupler than dinitrophenol. When administered in high doses, the drug aspirin acts as an uncoupler. Physiological uncouplers, certain physiological substances which act as uncouplers of higher concentration have been identified. These include the hormone thyroxine, long-chain fatty acids. The unconjugated bilirubin is also believed to act as an uncoupler. This is, however, yet to be proved beyond doubt. Significance of uncoupling. Uncoupling of respiration from oxidative phosphorylation under natural conditions assumes biological significance. The maintenance of body temperature is particularly important in hairless animals, hibernating animals, and the animal is adapted to cold. These animals possess a specialized tissue called brown adipose in the upper back and neck portions. The mitochondria of brown adipose tissue are rich in electron carriers and are specialized to carry out oxidation uncoupled from phosphorylation. This causes liberation of heat when fat is oxidized in the brown adipose tissue. The presence of active brown adipose tissue in certain individuals is believed to protect them from becoming obese. The excess calories consumed by these people are burnt and liberated as heat instead of being stored as fat. Ionophores. The term ionophores is used to collectively represent the lipophilic substances that promote the transport of ions across biological membranes. All the uncouplers described above are in fact proton ionophores. Ionophores. The antibiotics validomycin and nigersin act as ionophores for potassium ions. Both these compounds are also capable of dissipating proton gradient across the inner mitochondrial membrane and inhibit oxidative phosphorylation. The other inhibitors of oxidative phosphorylation are oligomycin. This antibiotic prevents the mitochondrial oxidation as well as phosphorylation. It binds with the enzyme ADP synthase and blocks the proton H+ channels. It thus prevents the translocation and re-entry of protons onto the mitochondrial matrix. Due to this, protons get accumulated at higher concentration in the intermembrane space. Electron transport, respiration ultimately stops. Since protons cannot be pumped out against steep proton gradients, the action of oligomycin indicates that oxidation and phosphorylation are tightly coupled processes. Atractyloside. This is a plant toxin and inhibits oxidative phosphorylation. And this is done by an indirect mechanism. The presence of adequate concentration of ADP and PI in the mitochondrial matrix and the transport of ATP into the extramitochondrial system are needed for phosphorylation to proceed continuously. Adenucleotide carrier system facilitates the transfer of ATP and ADP. Atractyloside inhibits adenucleotide carrier and thus blocks the adequate supply of ADP, thereby preventing phosphorylation. Transport of reducing equivalents, that's by the shuttle pathways. The inner mitochondrial membrane is impermeable to NADH, therefore the NADH produced in the cytosol cannot directly enter the mitochondria. Two pathways, namely glycerol phosphate shuttle and malate aspartate shuttle, are operated to do this job. They transport the reducing equivalents from cytosol to mitochondria and not vice versa. The shuttle pathway are not operative to the same extent in all the tissues. Malate aspartate shuttle predominantly occur in liver and heart, whereas glycerol phosphate shuttle operates in muscle and brain. and the glycerol phosphate shuttle cyanide. The systolic glycerol 3-phosphate dehydrogenase oxidizes NADH to NAD+. The reducing equivalent are transported through glycerol 3-phosphate into the mitochondria. Glycerol 3-phosphate dehydrogenase present on outer surface in a mitochondrial membrane reduces FAD to FADH. Dihydroxyacetone phosphate escapes into the cytosol, and the shuttling continues as depicted in figure 12. FADH2 gets oxidized via EDC to generate 2-ATP. Malate aspartate shuttle. In the cytosol's Xl-acetate accepts the reducing equivalents NADH and becomes malate. Malate then enters mitochondria where it is oxidized by a mitochondrial malate dehydrogenase. In this reaction, NADH and oxaloacetate are regenerated. NADH gets oxidized by electron transport chain and 3-ADP are produced. This is in contrast to glycerol phosphate shuttle, where only 2-ADP are produced. In the mitochondrial, oxaloacetate participates in transamination reactions with glutamate to produce aspartate, and alpha-ketoglutarate aspartate enters the cytosol and transaminates with alpha-ketoglutarate to give oxaloacetate and glutamate. Malate aspartate shuttle is shown in figure 13. And the next this topic will be coupled reactions in high energy compounds, energy-rich compounds. Now, metabolism is essentially a linked series of chemical reactions that begins with the particular molecule and converts it into some other molecules in a carefully defined fashion. There are many such defined pathways in the cell, and we will examine a few of them in some detail later. These pathways are interdependent, and their activity is coordinated by exquisitely sensitive means of communication in which allosteric enzymes are predominant. We can divide metabolic pathways into two broad classes. One is those that convert energy into biologically useful forms, and two, those that require inputs of energy to proceed. Although this division is often imprecise, it nonetheless is a useful distinction in an examination of metabolism. Those reactions that transform fuels into cellular energy are called catabolic reactions, or more generally catabolism. Fuels, carbohydrates, fats undergo catabolism to carbon dioxide, water, and useful energy. Those reactions that require energy, such as the synthesis of glucose, fats, or DNA, are called anabolic reactions or anabolism. The useful forms of energy that are produced in catabolism are employed in anabolism to generate complex structures from simple ones, or energy-rich states from energy-poor ones. Useful energy plus small molecules undergoes anabolism to complex molecules. It's broken up, it's built up. Some pathways can be either anabolic or catabolic depending on the energy conditions in the cell. They are referred to as amphibolic pathways. Thermodynamically unfavorable reaction can be driven by a favorable reaction. How are specific pathways constructed from individual reactions? A pathway must satisfy minimally two correct criteria. One is the individual reactions must be specific, and the entire set of reactions that constitute the pathway must be thermodynamically favored. A reaction that is specific will yield only one particular product or set of products from its reactants. The function of enzymes is to provide this specificity. The thermodynamics of metabolism is most readily approached in terms of free energy. A reaction can occur spontaneously only if delta G, the change in free energy, is negative. Recall that delta G for the formation of product C and D from substrates A and B is given by delta G zeros prime plus RT standard logarithm C and a D by A and a B. And thus the free energy of a reactant depends on the nature of the reactant and products expressed by the delta G0 term, the standard free energy change, and on their concentration expressed by the second term. All right. So an important thermodynamic factor is that the overall free energy for a chemically coupled series of reactions is equal to the sum of the free energy changes of the individual steps, consider the following reactions. A reversibly to B plus C gives delta G 0 prime equals plus 5, B to D at minus 8 kilocalorie, and finally A reversibly to C and D, delta G 0 prime given minus 3 kilocalories moles raised to minus 1 or minus 13 kilojoules moles raised to minus 1. Under standard conditions, A cannot be spontaneously converted into B and C because dG is positive. However, the conversion of B into D under standard conditions is thermodynamically feasible because free energy changes are additive. The conversion of A to C and D has a delta energy 0, that is free energy of minus 3 kilocalorie moles raised to minus 1 or minus 13 kilojoules mole raised to minus 1, which means that it can occur spontaneously under standard conditions. Thus, a thermodynamically unfavorable reaction can be driven by a thermodynamically favorable reaction to which it is coupled. In this example, the chemical intermediates B, common to both reactions, couples the reaction. Thus, metabolic pathways are formed by the coupling of enzyme-catalyzed reactions such that the overall free energy of the pathway is negative. ATP is the universal currency of free energy in biological systems. So, just as commerce is facilitated by use of common currency, the commerce of the cell metabolism is facilitated by the use of a common energy currency, adenosine triphosphate ATP, part of the free energy derived from the oxidation of foodstuffs and from light is transformed into this highly accessible molecule, which acts as the free energy donor in most energy requiring processes such as motion, active transport, or biosynthesis. ATP is a nucleoside consisting of an adenine, a ribose, and a triphosphate unit. The active form of ATP is usually a complex of ATP with magnesium 2 or manganese 2. In considering the role of ATP as an energy carrier, we see that ATP is an energy-rich molecule because its triphosphate unit contains 2 phosphoanhydrate bonds. A large amount of free energy is liberated when ATP is hydrolyzed to adenosine diphosphate and orthophosphate P1, or when ATP is hydrolyzed to adenosine monophosphate AMP and pyrophosphate BP1. So ATP plus water reversibly to AMP plus pyrophosphate 1, but free energy equals minus 10.9 kilocalories. Right, and ADP plus water reversibly gives ADP plus P1, and delta is free energy is equal to minus 7.3 kilocalories mole raised to minus 1 or minus 30.5 kilocalories. The precise dG for these reactions depends on the ionic strength of the medium and on the concentration of magnesium and other metal ions. On the typical cellular concentrations, the actual dG for these hydrolysis is important, minus 12 kilocalorie mole raised to minus 1. The free energy liberated in the hydrolysis of ATP is harnessed to drive reactions that require an input of free energy, such as muscle contraction. In turn, ATP is formed from ADP and P1 when fuel molecules are oxidized in chemotropes or when light is trapped by phototropes. The ATP to ADP cycle is the fundamental mode of energy exchange in biological systems. Some biosynthetic reactions are driven by hydrolysis of nucleoside triphosphate that are analogous to ATP, namely guanosine triphosphate GTP, uridine triphosphate UTP, the cytosine triphosphate CTP. The diphosphate forms of these nucleosides are donated by GDP, UDP, and CDP. Enzymes can catalyze, and the monophosphate forms by GMP, UMP, and CMP. Enzymes can catalyze the transfer of the terminal phosphoryl group from one nucleotide to another. The phosphorylation of nucleoside monophosphates is catalyzed by a family of nucleoside monophosphate kinases. The phosphorylation of nucleoside diphosphates is catalyzed by a nucleoside diphosphate kinase, an enzyme with broad specificity. It is intriguing to note that Although all of the nucleotide triphosphates are energetically equivalent, ATP is nonetheless the primary cellular energy carrier. In addition, two important electron carriers, NAD plus and FAD plus, are derivatives of ATP. The role of ATP in energy metabolism is paramount. And the ATP hydrolysis drives metabolism by shifting the equilibrium of coupled reactions. And how does coupling to ATP hydrolysis make possible an otherwise unfavorable reaction? Consider a chemical reaction that is thermodynamically unfavorable without an input of free energy, a situation common to many biosynthetic reactions. Suppose that the standard free energy of conversion of compound A into compound B is plus 4 kilocalories per mole. A to B, delta G0 gives plus 4 kilocalories. The equilibrium constant KEQ of this reaction at 25 degrees centigrade is related to dG0 in units of kilocalories per mole. Thus, net conversion of A into B cannot occur when the molar ratio of B to A is equal to or greater than 1 to 1.1.5 in 10 raised to minus 3. However, A can be converted into B under these conditions if the reaction is coupled to the hydrolysis of ATP. The new overall reaction is A plus ATP plus H2O reversibly gives B plus ADP plus Pi plus H plus, and free energy is equal to minus 3.3 kilocalories. Its standard free energy change of minus 3.3 moles is the sum of the value of dG for the conversion of A to B plus 4 kilocalories and the value of dG for the hydrolysis of ATP. At a pH of 7, the equilibrium constant of this coupled reaction is given here, equivalent B by A into ADP into Pi by ADP is equal to 10 raised to 3.3. At equilibrium, the ratio of B to A is given by B by A is equal to K constant, ADP by ADP into Pi. The ADP generating system at cells maintains the ADP by ADP Pi ratio at a high level, typically of the order of 500 m raised to minus 1. For this ratio, B by A is equal to 2.67 into 10 raised to 500 is equal to 1.34 into 10 raised to 3, which means that the hydrolysis of ATP enables A to be converted into B until the B by A ratio reaches a value of 1.34 into 10 raised to 5. This equilibrium ratio is strikingly different from the value of 1.15 and 10 raised to minus 3 for the reaction A to B. In the absence of ATP hydrolysis, in other words, coupling the hydrolysis of ATP with the conversion of A into B has changed the equilibrium ratio of B to A by a factor of about 10 raised to 8. We see here that the thermodynamic essence of ATP's action is an energy coupling agent. Cells maintain a high level of ATP by using oxidizable substrates or light as sources of free energy. The hydrolysis of an ATP molecule in a coupled reaction then changes the equilibrium ratio of products to reactants by a very large factor of the order of 10 raised to 8. More generally, the hydrolysis of an ATP molecule changes the equilibrium ratio of a coupled reaction or sequence of reactions by a factor of 10 raised to 8n. For example, the hydrolysis of 3 ATP molecules in a coupled reaction changes the equilibrium ratio by a factor of 10 raised to 24. Right? And thus, a thermodynamically unfavorable reaction sequence can be converted into a favorable one by coupling it into the hydrolysis of a sufficient number of ATP molecules in a new reaction. It should also be emphasized that A and B in the preceding coupled reaction may be interpreted very generally, not only as a different chemical species. For example, A and B, for example, A and B may represent activated and unactivated conformations of a protein. In this case, phosphorylation with ATP may be a means of conversion into an activated conformation. Such a conformation can store free energy, which can then be used to drive a thermodynamically unfavorable reaction. Through such changes in conformation, molecular motors such as myosin, kinesin, and dianin convert the chemical energy of ATP into mechanical energy. Indeed, this conversion is the basis of muscle contraction. Alternatively, A and B refer to the concentration of an ion or molecule on the outside and inside of a cell, as in the active transport of a nutrient. The active transport of sodium and potassium across membranes is driven by the phosphorylation of the sodium-potassium pumped by ATP and subsequently phosphorylation. Structural basis of the high phosphoryl transfer potential of ATP, as illustrated by molecular motors and ion pumps. Phosphoryl transfer is a common means of energy coupling. Furthermore, the phosphoryl transferase is also widely used in the intercellular transmission of information. What makes ATP a particularly efficient phosphoryl group donor? Let's compare the standard of free energy of hydrolysis of ATP with that of phosphate esters such as glycerol 3-phosphate. As per the formula given here, ADP to water gives ADP plus Pi plus H plus, and delta free energy minus 7.3. Glycerol 3-phosphate plus water gives glycerol plus phosphate organic plus delta G gives minus 2.2 kilocals. The magnitude of delta G for the hydrolysis of glycerol 3-phosphate is much smaller than that of ATP, which means that ATP is much smaller than the classification of high energy compounds. There are at least 5 groups of high energy compounds: paraphosphate ATP, acyl phosphates, 1,3-biphosphoglycerate, ethyl phosphates, as example, phosphonopyruvate, thioesters, acyl-CoA, and phosphoguanidines, phosphocreatine. There are some more details on the high energy compounds, including high energy bonds present in each category. Now, high energy bonds. The high energy bonds, compounds possess acid and nitrate bonds, mostly phosphoanhydrate bonds, which are formed by the condensation of two acidic groups or related compounds. These bonds are referred to as high energy bonds. Since the free energy is liberated when these bonds are hydrolyzed, Lippmann suggests the use of simple squabble to represent high energy bond. For instance, ATP is written as AMP, squabble P, squabble P. Storage form of high energy phosphates: phosphocreatine, creatine phosphate, stored in vertebrate muscle and brain, is an energy-rich compound. In invertebrates, arginine phosphate replaces phosphocreatine. Looking at this table, it shows that the high energy compounds: paraphosphate, ADP and paraphosphate, acyl phosphates, 1,3-biphosphoglycerate, enophosphates, phosphoenopyruvate, thiolesters, acyl-CoA, guanidine phosphates, phosphocreatine. Right? And next topic would be biological energy transducers and photosynthesis. Photosynthesis is essentially the only mechanism of energy input in the living world. Photosynthesis, photolights and synthesis, putting together, is anabolic process. It's a build-up process, anabolism, of manufacture of organic compounds inside the chlorophyll containing cells from carbon dioxide and water with the help of sunlight as a source of energy. A simple equation of photosynthesis is as follows. So that's 6CO2 plus 16H2O gives chlorophyll, CH2 plus C6H12O6 plus 6O2. And however, the function of water is to provide hydrogen or the synthesis of organic compounds. All the liberated oxygen comes from it. Therefore, the equation is modified as 6CO2 plus 12H2O given 6CO2, giving 6C6H12O6 plus, let's go again. A simple equation of photosynthesis as follows. 6CO2 plus 16H2O gives glucose, that is C6H12O6 plus O2, in the presence of chlorophyll and radiant energy. However, the function of water is to provide hydrogen for the synthesis of organic compounds, and all the liberated oxygen comes from it. Therefore, the equation is modified as chlorophyll. 6CO2 plus 12H2O gives C6H12O6 plus 6H2O plus 6O2. Chloroplast is the seed of photosynthesis and is best exemplified in the higher plants. The external surface of thylakoid contains the photosynthetic pigments and serves the ends of light reaction. The stroma, on the other hand, is concerned with the events of the dark reaction. Photosynthetic pigments. The photosynthetic pigments present in thylakoid membranes consist of largely of two kinds of green chlorophylls. Chlorophyll A, that is C55H70O5N4Mg, and chlorophyll B, C55H72O6N4Mg. Chlorophyll A and B, right? Also present are yellow to orange pigments classified as carotenoids. There are two kinds of carotenoids, the pure hydrogen hydrocarbon carotenes and the oxygen-containing xanthophyls. Certain carotenes, especially while xanthine, a xanthophyll also exists in the chloroplast envelope, giving it a yellowish color. In most plants, including green algae, beta-carotene and the lutein are the most abundant carotenes in the thylakoid. Photosynthesis consists of two types of reactions, a light-dependent one and a light-independent one. The light-dependent reaction is a photochemical reaction, right, or a light reaction as it came to be called, culminating in the generation of NADPH2, ATP, and evolution of molecular oxygen. The NADPH2 and ATP are energy-rich, having caught the electrons that became available when light impinged upon chlorophyll. They form the assimilatory power utilized for carbon dioxide fixation. The events of carbon dioxide fixation is light-independent reaction and is designated as dark reaction. Light reaction consists of two phases. Phase 1, energy absorption and retention of light by the photosynthetic pigments, and phase 2, energy transduction conversion of light energy absorbed in phase 1 into chemical energy ATP and NADPH2 by photophosphorylation. More green plants absorb light in the visible spectrum 390 to 700 nanometer, whereas purple bacteria employ wavelengths ranging from near ultraviolet to infrared 800 to 950 nanometers. This range of the spectrum through which photosynthesis can take place is called photosynthetically active radiation. But the entire range is not employable in photosynthesis. The green plants, for example, absorb light maximally in the red and blue regions of the spectrum. A study of the absorption spectra shows the quantitative relationship between the wavelength of light and its absorption by the pigment in question. Thus, we see the chlorophyll A has its absorption peaks at 660 nanometers and 430 nanometers, chlorophyll B at 648 nanometers and 456 nanometer, carotene at 478 nanometers and 449 nanometers, and xanthophyll same as carotene. And photosynthesis. The photosynthesis has been located in the thylakoid membranes. It is made up of three forms of chlorophylls A, one absorbing maximally at 683 nanometers, and the second absorbing maximally at 695 nanometers, and the third at 670 nm. The last of these has been called P700. Photosystem 2 has been located in the stroma thylakoids. It is made up of two forms of chlorophyll A with maximum absorption at 670 and 690 nanometers. The second form is crystalline P690. Each photosystem has three components. One, a reaction center made up of a special chlorophyll molecule in the photosystem 1. It is a protein-bound chlorophyll molecule, P700. In photosystem 2, it is 690. The reaction centers are the actual sites where the light energy is converted to chemical energy. 2, some electron carriers in photosynthesis 1, 10, plastocyanin. Photosystem 1, 10, plastocyanin, cytochrome F, and ferrodexin as the electron carrier. Photosystem 2 has plastoquinone and cytochrome B559. Third would be other chlorophyll and carotenoids, which merely serve to transfer the light absorbed by them to the active centers. Photosystem 1 takes part in both cyclic and non-cyclic photophosphorylation. PS1 can carry on cyclic photophosphorylation independently. Normally, it drives an electron from photosystem 2 to NADP, plus photosystem 2 picks up electron. It's released during photolysis of water. The same is extruded on absorption of light energy as electron passes over cytochrome complex. Sufficient energy is released to take part in synthesis of ATP from ADP and inorganic phosphate. This photophosphorylation is non-cyclic. PS2 can operate only in a conjunction with PS1.